Hello, I'm Chris.
And I'm Katie.
Today we're going to briefly discuss what we did over the summer at Hamline University. We used CRISPR-Cas technology to knock out selected genes in a model organism known as Arabidopsis. It's a very small weed-like plant, and we'll get into more detail what that plant is and what we did in the near future on this video.
So to start off with, I'm going to mention just a little about what CRISPR-Cas9 is. So it uses this paired mechanism. It uses an enzyme called Cas9, and then it uses guide RNA to make a double-stranded break in the genome of an organism so that DNA then can be added or removed. And we'll show you a little bit more on that later as well. Yeah, so Chris mentioned a little bit about this Arabidopsis plant in the intro over there, and so now I'm just going to talk a little bit more about them. We used these tiny, cute little seeds, and we planted them using a micropipette, and basically we just squeezed up the seed with a little bit of water, and we planted one in each pot, and we had 20 plants to each bin, and then we would put them in storage, and then essentially we would grow them in a growth chamber. And then about a month and a half later, these cute little seeds would become plants that had flowers, and that meant that they were ready to be transformed. So next, we're going to talk a little bit about the plasmid vectors that we used throughout this whole process. We had vector A, which contained the Cas9 enzyme used to cut out the genes in the transgenic plants. We had vector B, which was the first vector that we had to create, and we'll talk a little more in depth about that in just a few moments. But essentially, it contained the genes that were to be knocked out. We had vector C, which was an empty vector. You can place whatever else you want in it for your transgenic plants, but we just used it as merely an empty vector. And then we had vector D, which was the transformation backbone that all three of the other vectors went into, and then that was inserted into the plants.
So, to expand a little bit more about vector B, what Katie just stated, the ones we targeted were ARC, which was having the plant's roots become twisted and knotted. The AT gene, which was having the plants become yellow at maturity. PPL was having the plants have different shaped leaves than what they normally would have. DWF is dwarfism of the plant, so they come out even smaller than normal. And ERD is cold resistance. So what we did with these targeted genes is that we took out the CCBD portion of the vector, and we replaced it with these targeted genes. So you can see right here, we replaced it with what we wanted to have become replicated in the bacteria, which was then added to the plants during transformation.
So here we wanted to just show a little diagram of how the three vectors went into the big backbone. We use this process called Golden Gate reaction, which essentially just cuts and pastes various portions that you want of vectors into one big one. And we used this restriction enzyme AARI, which made two cuts in a vector, and then we took the region between those two cuts that we wanted, and then we put it into the big backbone.
Now that we have successfully transformed vectors as well as mature enough plants, we can go ahead and transform our plants with the bacteria. This is known as floral dipping. Once the plants have been successfully infected by the bacteria, we wait for them to grow seeds, which we can harvest. Then we take those seeds and we place them on an agar plate, and the ones that grow on there we know are successful with the transformation. Now that we discussed the method, it makes sense just to show you a few pictures when we actually did it. We can see the day one right before they're about to get dipped, during the dipping, and when we pulled them out, they look pretty sad. But that's okay, because after 3 days they start growing and they're happy again with brand new flowers.
So next, I just wanted to show you the process that we used for verification while we were doing all of these Golden Gate reactions and other various steps. So when we made our V vector, we ran a gel electrophoresis, and then we did Sanger sequencing just to show that our fragments were approximately the right size, and then to show that within our fragments, the important sections matched our designed vectors that we made, and then we did the same thing when we made our final vector.
Congrats, you made it to the end of our video. Before we go, we'd like to summarize the key things that we did over the summer one more time. We grew Arabidopsis plants. We used the Golden Gate reaction to create vectors, which we used to place into bacteria. From there, we took that bacteria and we infected our plants so that the genes we wanted to be knocked out were actually knocked out. All around, it was a very successful summer. Thanks for watching. See ya!
See ya!